Am I audible?
Yes, sir.
Yes,
Yes,
sir.
sir.
Okay, okay.
Yeah, sir, audible, clear.
Good evening, sir. This is Manjula.
Madam Ji, good evening. My dear friends, good evening. Nice to meet you.
Thank you,
Very
sir.
nice evening, my dear friends.
Thank you, sir.
Now
Yeah.
I'm sharing my screen. I hope my screen is visible.
Yes, sir.
Yeah. This cyanobacteria is visible.
Yeah, sir, it is visible.
The definition, classification is visible.
Yeah, sir, everything
Yes,
is
Yeah.
sir. Yes, sir.
Okay, good, my dear friends. A wonderful evening, my dear friends. And today is the first class in faculty exchange program. We are meet here. So both the colleges, that is the Women's Degree College, Government Degree College for Women, Jagtial, and one of the prestigious institution in Twin Cities, that is the Rbvrr Women's College. Both they are mingled, and they are exchange the faculty among our students, sharing ideas, knowledge, or experience. So that's why we are meet here. So this is a wonderful opportunity, my dear friends. I hope you clearly listen to. If you have any doubt during my class, please ask me. I will clarify your doubts, no doubt at all. My dear friends, this is Padala Tirupathi from College for Women Jagtial. I am working as an assistant professor, Department of Botany of Government Degree College for Women Jagtial. So today we are going to discuss about one of the most important area in algae, that is a blue-green algae. Have you ever heard the term blue-green algae?
Yes, sir.
Yeah. So blue-green algae, one of the primitive algae, one of the primitive and ancient algae, which is the prokaryotic living organism. My dear friends, remember, blue-green algae is the first algae, which is the entire earth. Which is the first living organism in the entire earth is blue-green algae. Remember, blue-green algae. So this blue-green also we can call as cyanobacteria or mixophyceae or cyanophyceae. Come on, anybody can repeat. This is the cyanobacteria we can also call as,
Cyanophyceae.
Mixophyceae,
Mixophyceae.
Cyanophyceae,
yeah, cyanophyceae, mixophyceae,
Mixophyceae,
blue-green algae, that is
algae.
BGA. Okay, and compared to other algae, so this blue-green algae belongs to a prokaryotic organism, prokaryotic living organism. Actually, algae, all the alga organism are mainly they are belongs to eukaryotic organism, but our blue-green algae, cyanobacteria, is belongs to prokaryotic organism. Prokaryotic organism. My dear friends, this is autotrophic thallophytic organism. Autotrophic thallophytes. You know the term autotroph.
Yes, sir.
Yeah.
Yes, sir.
What do you understand
They
about
prepare own food.
Yeah, yeah, very good, excellent, my dear friend. So they prepare their own food material. They are not depend any other organism. They are capable for the production or prepare their own food material in the process of photosynthesis. So that's why this cyanobacteria or alga we can also call as autotrophs. And the plant body belongs to thallus. So have you ever heard the term thallus?
Yes, sir.
Yes,
Yeah,
sir.
sir,
What do you
I
It
understand?
is not differentiated into
Undifferentiated.
stem,
root,
root,
stem,
leaf.
and root.
Yeah, stem, leaf, root. These are the parts we know. These are the consist in all plant parts. But in our algae, even though fungi also, in algae along with fungi, they are not differentiate into root, stem, leaf.
Stem,
So
leaf.
the third plant body we can call as thallus. So this cyanobacteria is also autotrophic thallophytic organism. Okay, and this photosynthetic organism which release the an electron donating substrate. Have you any doubt, Amma? My dear friends, have you any doubt?
No, sir.
No, sir.
Good, good. So these the classification of the cyanobacteria. We know that the kingdom Monera. Have you ever heard term Monera?
Yes, sir.
Yes, sir.
Yeah, sir.
So according to RH Whittaker, entire organism has classified into 5 kingdoms. How many kingdoms? 5.
5 kingdoms.
The
5
first kingdom,
kingdoms.
Indic kingdom and ancient kingdom, the first kingdom is Monera. Anybody can remain tell some? Anybody?
Protista, Monera,
Yeah,
Protista, Plante, Animalia.
good, excellent. So
Sunlight.
these are the 5 kingdoms RH Whittaker has classified. So these, according to RH Whittaker, so Monera is the first kingdom. So our cyanobacteria belongs to kingdom Monera. And division, Eubacteria. Class Cyanobacteria. Some of the examples related of the cyanobacteria is Nostoc, Oscillatoria, Anabina, Spirulina, Lingbaia, Stegonema, Azolla, Gliotrichia. So these are the some of the examples related to our cyanobacteria. So is there any doubt in this slide, Amma?
No, sir.
No, sir.
Then we will going to discuss about the what are the characteristic features of cyanobacteria or blue-green algae or mixophyceae or cyanophyceae members. So cyanobacteria are prokaryotic. What do you understand about the prokaryotic, Amma? Anybody?
It is not well developed nuclear membrane.
Nucleus is not
Good,
well developed.
good. So two answers. So
Incipient, sir.
both answers are correct. Remaining? Anybody? So this
Not develop the cell wall.
cell wall is present, Amma, my dear friends. I am asking.
Incipient.
Caryo means nucleus. Caryo means nucleus. Caryo means nucleus. Pro means before or primitive. So before the formation of the nucleus or the nuclear membrane. So this prokaryotic means before eukaryotic living organs are not a distinct nucleus is present in these type of organism. Maybe the unicellular forms, maybe the multicellular. There is no definite nucleus is present in understand about aerobic.
Yes, sir.
They
Presence
are
of air.
Yeah, they are utilizing oxygen in the
Presence of
process.
oxygen.
Yes, they need oxygen in the process or other living or other physiological functions. And photoautotrophic organism. Photoautotrophic means they are prepared their own food material in the presence of
Presence of
light.
light.
Yes, good.
Presence of sunlight.
Light.
Yes, presence of sunlight, definitely. So that's why the what do you understand about this first point? Cyanobacteria, prokaryotic and photoautotrophic organism. In this photoautotrophic organism, they are depends for the carbon source from carbon dioxide in the atmosphere. Have you ever heard about the carbon dioxide? The term carbon dioxide?
Yes, sir.
Yes, this is the gas. This is the gas which is present in the atmosphere. How much percentage? 0.03%. So this carbon dioxide. So these photoautotropes, they are depends upon the for carbon from carbon dioxide of the atmosphere. Then they are depends upon the energy from sunlight. Is it clear? So that's why we are called photoautotrophic. We know that we heard sometimes chemoautotrophs. Have you ever heard term
Yes, sir. It prepares
chemoautotrophs?
own food in presence of chemicals.
Yes, very good, my dear friends. So that's why we can call chemoautotrophs. Here, our blue-green alga, they are prokaryotic, aerobic, photoautotrophic organism. Here, some of the friends are already told that prokaryotic means does not possess a definite nucleus. But
Nuclear membrane.
yes, definite nuclear membrane. Along with this prokaryote, they are does not possess any unit membranous cell organelles. They are not present in the cell. Like like mitochondria, like Golgi apparatus, like endoplasmic reticulum, like plastids. These type of cell organelles which does not possess in prokaryotes. Am I clear?
Sir.
That we can also call membrane-bound organelles are absent in cyanobacteria. Is it clear?
Yes, sir.
So the range, the size of the cyanobacteria ranges from 1 millimicron to 10, 1 micrometer to 10 micrometer range. It contains primitive nucleus. That's why we can all on membrane-bound cell organelles. They are not present in this cell of the prokaryotes or cyanobacteria. They are endoplasmic reticulum here, Golgi apparatus, mitochondria, like
Plastids.
chloroplast. Plastids, very good. So these cell organelles does not possess in this prokaryotes. Then in this cyanobacteria, they are possess the pigments. They are the one of the most important aspects for the preparation of the food material. That is chlorophyll A. Have you ever heard term chlorophyll A?
Yes, sir.
Chlorophyll A is the main
Pigment.
important, the first important and the very important. All photosynthetic organism, they are definitely possess this chlorophyll A. Chlorophyll A is act as a reaction center or light harvesting complex. In that light harvesting complex, chlorophyll A is the reaction center. So one of the important pigment, chlorophyll pigment, that is chlorophyll A. So among in our algae, we know that I hope you already studied or are going to study about the pigments chlorophyll A, chlorophyll B, chlorophyll C, chlorophyll D, chlorophyll E also present. But in our cyanobacteria, chlorophyll A is only present. Remaining chlorophylls, they are not present in the cyanobacteria. Chlorophyll A, along with this chlorophyll A, other phycobilins like phycocyanin C and picoerythrin C, or C phycocyanin or C picoerythrin. So these are the pigments which are present in cyanobacteria members. How many types of the pigments we are observing in cyanobacteria, my dear friends?
3 types of pigments.
Chlorophyll A, phycocyanin, picoerythrin.
Erythrin.
Okay, so these are the three pigments which they are the responsible for the pigmentation or color of the cyanobacteria members. Chlorophyll A responsible for which color, my dear friends?
Green
Green
color.
color.
Yeah. Then whereas phycocyanin,
Blue color.
Blue color.
Blue
then,
color.
Blue color, sir.
then phycoerythrin.
Red
Red
color.
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 Red color.
My dear friends, remember here, erythrin. We know that erythrocytes.
RBC.
Yes, good, excellent, my dear friend, excellent. So phycoerythrin, phyco means plants. They are present in the plant, especially in cyanobacteria, cyanophysin members. So phycoerythrin is responsible for the red color. Okay, then phycocyanin is in parts blue color. Then chlorophyll A belongs to green color. So these colors are mingled. The cyanobacteria members, the color is appearance our naked eye, our eye. These members, these plant body and the color is which color, my dear friends? Blue green color. BGA. So this BGA, blue green alga, which is responsible, which type of pigments are responsible? No, please silence.
Phycocyanin and chlorophyll A.
Yeah, and phycoerythrin also. So so these are due to the chlorophyll A and phycocyanin. Very good. So chlorophyll involves in photosynthesis, especially photosystem 1, whereas phycocyanin is involved in photosystem 2. So its photosynthesis is oxygenic. So cyanobacteria have the ability to fix atmospheric nitrogen. The reserve food material is cyanophysin starch. So in this slide, what we are discussing about? So the size of the cyanobacteria members and which type of pigments are responsible for the blue-green algae, especially these
It's not.
pigments, chlorophyll and
Sir, we
phycocyanin.
are unable to understand what you are
Sir,
saying,
we
sir.
are unable to understand. It's not audible. Some
The
disturbance is
technical
coming.
disturbance is coming from your mic.
My mic.
And double sound is
Yes,
also
sir.
coming, sir.
Lot of noise is coming.
Okay, I will stop. Now it's visible. Am I audible?
Yes,
Yes,
sir.
sir.
Yes, sir.
Yes, sir.
Yes, sir.
Okay, good, good,
Yes,
my dear
sir.
friends. Okay, so blue-green alga. Is it clear, my voice?
Yeah, sir. It's audible.
Yes,
సార్
sir.
సార్
Everything
ఎక్స్క్యూజ్
is
మీ
Yes,
వన్
sir.
okay,
మినిట్
Yes,
okay,
sir.
సార్
sir.
సార్
Okay,
మీరు
good.
టూ డివైస్ నుంచి జాయిన్ అయినట్లున్నారు డిస్టర్బెన్స్ వస్తుంది
Oh
సార్
my.
సారీ ఇస్ లెఫ్ట్ నో గర్ల్స్
Yeah, ma'am.
Am I audible?
ఓకే
Yes,
సార్
sir.
మీరు
Yes, sir.
Okay, right. So our blue-green algae contain chlorophyll A, phycocyanin, and phycoerythrin. Chlorophyll A. So during my class, my dear friends, all of you, please mute yourself. Rajini Madam Garu, you're also. Please mute yourself, all of you. Okay. So during this photosynthesis, so chlorophyll A is act as photosystem 1, along with this phycocyanin is act as photosystem 2. And this photosynthesis is oxygenic, which means which liberates during the photosynthesis this organism of cyanobacteria which liberates oxygen. Then cyanobacteria is also, along with this photosynthesis, one of the physiological function. Along with this photosynthesis, cyanobacteria is also responsible for the fixation of the atmospheric nitrogen. So this is the most important area for the fixation of the nitrogen from atmosphere to soil. So that's why these cyanobacteria members is also used as biofertilizers. Very, very important, my dear friends. And the reserve food material of this cyanobacteria is cyanophycean starch or myxophyceous starch. Myxophycean starch or cyanophycean starch. This is the reserve food material. This is the reserve food material. And then afterwards, then what is the habitat of this blue-green algae? The habitat of this blue-green alga is this not a similar a habitat, but most they are genetically diverse organisms that show a broad range of habitats. So they can live freshwater, they can also live in marine water, they can also live in damping places and moist soil. And one of the most important point is this blue-green alga is also able to survive in the hot springs like more than 80 degrees Celsius range. Conditions like hot springs is also suitable for the growing of this blue-green algae. So the habitat of this blue-green alga is freshwater, marine water. Freshwater means pond, lake, okay, and water. So marine water, which is the best example for the oceans, seas, and damp places. Damp places means where the water is going to, the water bodies, they are in the stable water or stagnant water, and the moist soil. Moist soil means the shady and moist soil, which is also suitable for the growing of this blue-green algae. And along with these conditions, the extreme conditions like hot springs is also suitable for growing of this blue-green algae. Then going to the next area, that is the thallus.
Then
Thallus. Type of the thallus. Which type of thallus we are observed in this blue-green alga? So these blue-green alga members, they are maybe unicellular, maybe multicellular, and colonial forms are also there. My dear friends, please mute yourself. All the members, please, all the students, please mute yourself. Then the unicellular organism, multicellular organism, and the colonial forms, then filamentous forms also we are observed in the cyanobacteria members. So the best examples of this unicellular is Crococcus. So this Crococcus is the body, the plant body, or the blue-green alga or the cyanobacteria member, which possess only single cellular. The single cell is also have the ability of the all types of the functions like reproduction, like digestion. So whatever it may be. So these type of functions which are made are in this single cell. So the best example of this unicellular cyanobacteria member is Crococcus. And this thallus having the mucinous sheath covering on the surface of the thallus. And the colonial forms. Colonial forms, these are the multicellular forms. It forms when a single cell's colonies are themselves, my dear friends. So the best examples of these colonial forms of the blue-green alga or cyanobacteria are Glyocapsa. So Glyocapsa is the best example for the colonial forms of the cyanobacteria members. Then majority of the cyanobacteria members or majority of the blue-green alga members, they are filamentous forms. So the filamentous forms, in these filamentous forms, number of cells or number of cells which are arranged in a beaded nature, in a beaded structure, and this beaded structure is covered by a mucilaginous sheath. So that we can also call as trichomes, or we can also call as the filamentous forms. So these filamentous forms, it consists, or the filaments also it consists heterocysts. So heterocyst is also one of the most important part of the filaments or trichome. So this heterocyst is also responsible for the fixation of the atmospheric nitrogen into the this plant body or the thallus. Okay, and this structure of this heterocyst compared to other cells of this trichome, other cells of this filament body, the heterocyst cell is larger than the other cells, and it helps to fixation of the atmospheric nitrogen into the soil. Then akinete. So akinete is also important part of the trichome or filamentous. So this akinete is responsible for the reproduction, and it also responsible for the collects of the food material. Best example of the filamentous forms are nostoc and oscillatoria. And remember, my dear friends, this filament is also, my dear friends, again classified into based on the whether the branches are present or the branches are absent. So the filament may be the branched filament, or the filament, the branches are not possessed in this filament. We can also call as unbranched filament. Unbranched filament. So branched filament or unbranched filament is also we are observed. Okay, so this branched filamentous structure, the best example of this blue-green alga is Cytonema and stegonema also. Some of the pseudo branches also we are observed in these filamentous forms of the cyanobacteria members. Anybody respond? Only one
Sir,
person.
voice breakage, sir.
My voice is breaking. Now it's okay.
Yes, sir.
Yes, sir.
Okay. Please mute yourself. Is there any problem? Please ask me. Okay, then we will move for next slide. So in this slide, we are observed the beautiful structure of the whether the members, whether they are the unicellular forms, whether they are in the colonial forms, whether they are in the filamentous forms. Is it clear? This the structure of the cell is visible. Anybody? One person respond, please.
Yes, sir.
Okay, good. Please mute yourself. Then, so this is the unicellular member, and this unicellular membrane is surrounded by a thick mucilaginous sheath, my dear. Thick mucilaginous sheath is responsible for the survival of the survival from the extreme conditions like hot springs. Okay, the heat, the hot condition is also suitable for the growing of these cells or the thallus of this cyanobacteria, just because of they possess thick mucilaginous sheets. So this is the unicellular forms, and this is the multicellular forms, and this is the filamentous form of the cyanobacteria. This filamentous forms of the cyanobacteria consist a beaded-like structures. They are arranged in the chain-like manner. So these are the cells. They are arranged in a beaded manner, and in this chain-like structure is further surrounded by a thick mucilaginous sheath. So this entire structure we can also call as trichome. So these filamentous form may be branched or unbranched. Among these cells, larger cells we are observed among these cells. So the largest structure we are observed, the polar nodules we are observed in this heterocyst. The heterocyst is responsible for the fixation of the atmospheric nitrogen in the thallus. And this is the one cell of the multicellular form. So this is the cells we are going to explain here. This, I hope this entire structure you are observed. So this is the cell. This is the cell which cell is possess a thick mucilaginous sheath. We are observed the yellowish color. This yellowish color mucilaginous sheath is a thick mucilaginous sheath is we are observed. This mucilaginous is made up of pectin, my dear friends. Pectin. This mucilage is processed pectin. Whenever this pectin is going to dissolve in the water, this surface of the cell wall or surface of the trichome or surface of the thallus is going to touch. Whenever we are going to touch, this entire nature is the mucilaginous nature. Just because of pectin is going to dissolve in the water, and it we are going to touch it looks like a mucilaginous. Okay, so this mucilaginous sheath is responsible for the growing of the extreme conditions like hot water and hot springs more than 80 degrees Celsius. And afterwards, this mucilaginous cell wall, then another cell wall, another wall we are observed in this cell wall. My dear friends, remember any plant cell wall is also the chemical composition of the cell wall. Cell wall possess which type of chemicals or which type of chemical component we are observed in cell wall? Our plant cell wall is made up of
Cellulose, chitin.
Good. Please mute yourself. So then this blue-green alga cell wall is made up of what we can say that murin or mucopolypeptides. Okay, so these mucopolymeric, so mucopolymeric cell wall we are observed. So this muramic acid. So my dear friends, along with this blue-green alga, so bacterial cell wall is also made up of murin or mucopolypeptide. So this mucopolypeptide or murin is also we can call as muramic acid or glucosamine. So glutamic acid, So these are all included in mucopolymeric wall. And then afterwards, the plasma membrane or cell membrane is also we are observed. So this is the cell membrane. Any plant cell or any alga membrane is also the cell wall. After cell wall, so the beneath the cell wall, plasma membrane we are observed. So the plasma membrane also we can call as cell membrane. So cell membrane is made up of lipoproteins or lipoproteins or lipoprotein layer. Plasma membrane is made up of lipoproteins or lipoproteins or proteins and lipids. Okay, please mute yourself, my dear friends. Please mute. Then the inside the cell we are observed no membrane bounded cell organelles are present. So they are absent in prokaryotic cell because this is the prokaryotic cell. They are not present in this cell. Only the very primitive cell organelles, very primitive cell organelle, my dear friends, that is ribosomes is present. Ribosomes are present. So these ribosomes belongs to 70th type of ribosomes. 70th type of ribosomes, they are present in this cell. You know that 70th ribosomes and 80th ribosomes are present in prokaryotes, whereas 80th type of ribosomes are present in eukaryotic cell. So our cyanobacteria cell are prokaryotes, that's why the ribosomes are 70th type of ribosome. And these ribosomes possess two units, that is the smaller subunit and the larger subunit. The larger subunit is 60s substance, 50s, and the smaller subunit is 30s. So 30s and 50s, these are the two subunits of the 70s type of ribosomes. So my dear friends, remember the very primitive cell organelle is ribosome. So these ribosomes is responsible for synthesis of the proteins. Ribosomes are responsible for the synthesis of the proteins. So this cell is also possess along with this thylakoid membranes also present. So the thylakoid is responsible for the photosynthesis. They have pigments like chlorophyll A and other pigments like cyanophysin, picocyan and picoerythrin. So these are the pigments which are present in the thylakoid membranes. And the nucleoid, the nucleoid structure we are also observed in this cell. The nucleoid means they are in the place of the nucleus, but they are not surrounded by a specific definite nuclear membrane. So that's why the DNA of this cyanobacteria we can call as a nucleoid. So along with this the cyanophysi member, or along with this cyanobacteria, along with this nuclear circular DNA structures we are also observed in this cyanobacteria. Circular DNA structures also we are observed in this cyanobacteria and also bacteria. Okay, so the storage food material, the stored food material of this cyanobacteria is cyanophysian starch. Cyanophysian starch. So this is about the structure of the cell. This is about the structure of the cell. Now we are going to discuss about the this entire structure. So sheath, that is we can also call as that mucilaginous sheaths. So this envelope of the cyanobacteria mucilaginous means hygroscopic. Hygroscopic in nature means they are absorb the water. Hygroscopic means they are attract the water. It contains the pectic substance. Whenever this pectin is going to soluble with this water, then the entire sheet is going to a gelatinous nature. Is fibrillar under the microscope. The main function of this sheath is protects the cell from unfavorable conditions. We know that extreme conditions like more than 80 degrees Celsius, though the high regions, then very colder regions, very deeper oceans. Okay, so these are the unfavorable conditions or extreme conditions. Whenever unfavorable conditions are occur, or even though they are grown in the extreme conditions, this mucilaginous sheath is going to protect the cells of the cyanobacteria members, and it keeps the cell moist. Always this mucilaginous sheath is going to keep the cell is going to moist. Okay, then what about the cell wall? So it is present between the sheath and plasma membrane. The pores that are present on the cell wall helps the secretion of mucilage. It consists of 4 layered represented by L1, layer 1, layer 2, layer 3 and layer 4. So cell wall is having how many layers? 4 layers, and they are these 4, among these 4 layers, they are interconnects with each other by plasma desmata. So this plasma desmata is the connecting link between the these 4 layers of the cell wall. And the next point, that is the plasma membrane. The next part of the cell is the plasma membrane. So this plasma membrane is the bilayered membrane. So plasma membrane is the thickness of this plasma membrane is 70 angstrom units. 70 angstrom unit thickness we are observed. Then selective. My dear friends, remember all plasma membranes are selective permeable membranes or semi-permeable membrane. So this maintains the structural integrity of the cell. There is a structure called the lamellosomes. So these lamellosomes forms when the plasma membrane invaginates and fuses with the lamellae. It encloses cytoplasm and other cell inclusions. Then cytoplasm. So the cytoplasm, which is present inside the cell, so it consists of 2 regions. The center part of the cytoplasm we can call as centroplasm. Then whereas the peripheral area of the cell, the peripheral area are we can call as a chromoplasm. So these 2 regions we are observed in the cytoplasm. The chromoplasm, chromo means color, which is responsible for the pigmentation, which possess the pigments of different pigments like carotenoids or phycobilins and chlorophylls. So these are responsible for the photosynthesis or photophosphorylation process. So this is the main physiological function we are observed in the chromoplasm, which is arranged in the peripheral area of the cell. Whereas the centroplasm, which center means in the center part of the cell or in the middle part of the cell. So it is colorless, not colored. It is colorless and regarded as the primitive nucleus, that is devoid of nuclear membrane and nucleolus. Some granular structures also present in this area, that is the centroplasm. So centroplasm is the part whereas the nucleus is present, but in the blue-green algae they are not present in the cell. So that's why the centroplasm does not possess the nucleus, it is a primitive nucleus. We can also call it a primitive nucleus. The centroplasm is also the granular structures are present, whereas this chromoplasm, which is responsible for the coloration of the thallus and also which is responsible for the photosynthesis, for the preparation of their own food material. So which is arranged in the peripheral area of the cell. Okay, then cytoplasmic inclusion. What are the inclusions we are observed in the cytoplasm? So some of the inclusion are like cyanophysin granules. So these granules, cyanophysin granules, these are present in the chromoplasm. This is also present in centroplasm. So the cytoplasmic inclusions which are present in centroplasm. So it is the nitrogenous reserve material. It constitutes about 8% of the total cell weight. So cyanophysis granules are how much weight? 80% of the total cell weight. Then gas vacuoles. So these gas vacuoles is responsible for the free floating on the surface of the water. They are responsible for the preservation of the gases inside the thallus. So these are hollow, means a space structure, you know. So tough and elongated in shape. Its diameter is 75 nanometer and length is 200 to 1000 nanometer diameter. Okay, then carboxysomes. So these are polyhedral bodies which contains 1,5-ribulose bisphosphate carboxylase. Have you ever heard on this 1,5-ribulose 1,5-bisphosphate? This is the most important carboxysome which is present in. We know that C3 cycle or Kelvin cycle, which is the first absorbent. So first CO2 acceptor is 1,5-ribulose 1,5-bisphosphate. Okay, ribulose 1 RBP carboxylase. So RBP carboxylase is an enzyme. Carboxysome is an enzyme. So this ribulose 1,5-bisphosphate carbon also can call as Rubisco. So the entire plant world, Rubisco is one of the largest enzyme, one of the abundant enzyme, largest enzyme, and which is the dual nature of the enzyme. So dual nature of the enzyme, or bimorphic or dimorphic enzyme. So which is act as wherever the carbon dioxide is present. So this RUBP carboxylase is acts as carboxylase, whereas the carbon dioxide is going to decrease. The lesser amount of the carbon dioxide is available, or more than compared to carbon dioxide, oxygen level is increased. This ribulose 1,5-bisphosphate carboxylase enzyme is act as not carboxylase, which is act as oxidase. So that's why this enzyme we can call as carboxysome is act as it is a dimorphic enzyme, abundant enzyme, largest enzyme, and dimorphic enzyme. Very, very important. And the phosphate bodies. So phosphate bodies are nothing but violetin granules, my dear friends. So violetin granules which are present, which they have possess phosphates. Okay. Then phycobilisomes, very, very important, my dear friends. So phycobilins are the pigments, the special type of pigments which are present only red algae and blue-green algae, my dear friends. Red algae and blue-green algae. So these phycobilins, they are possessed in the phycobilisomes. So phycobiliproteins, when aggregates into a ball-like structures, form phycobilisomes. It is attached to the outer surface of the thylakoid. So we know that the cell possesses the thylakoid membranes. The thylakoid membranes also have the outer surface of the thylakoid membrane. They are possessed phycobilisomes. So phycobilisomes are proteins. They are phycobiliproteins. Phycobiliproteins. So when they are then aggregates into a ball-like structures, they are phycobilisomes. So phycobiliproteins also we can call as phycobilisomes when they are going to aggregate, and which are attached on which space? So outer surface of the thylakoid membrane. Outer surface of the thylakoid membrane. And along with these phycobilisomes, my dear friends, phycobilisomes are present only algae, especially red algae and blue-green algae. Okay. So the Red Sea. Have you ever heard term Red Sea? Samudram. So this Red Sea is also responsible. The coloration of the Red Sea just because of erythrin. Erythrin is the pigment which are present in red algae. So Phycodesmium erythrium. The erythrium which is responsible for the Red Sea. Then the DNA matrix. My dear friends, remember there is no definite nucleus is present in this cell of the so that's why DNA is a naked DNA. So naked DNA fibrils or DNA matrix we are observed. So that naked DNA fibrils are dispersed in the cytoplasm. And my dear friends, remember here there is no nucleoplasm. Why? Because there is no definite nuclear membrane are present, and so that's why there is no definite nucleoplasm. So which is also we can also call as nothing but lacks of the nucleoplasm. There is no nucleoplasm is present. Only DNA, naked DNA. So that is open DNA fibrils we are observed in the DNA matrix. The base composition of the DNA differs. The cyanobacteria contains covalently closed and non-functional circular plasmid DNA. So in the previous slide, what we observed in this, along with nucleoid, so the DNA circular structures we are observed in this cell, the circular DNA structure we can also call as plasmids. So these plasmids are very, very important for the color, for the especially DNA or DNA technology in the genetic engineering method or biotechnology. In the field of biotechnology, the uses this plasmid DNA used as a vector for the transmission of the new DNA, DNA transfer. Okay, so this is about the plasmid DNA. So if you have any doubt, please ask me about today's class. Today we are already discussed about the general characteristic features of the cyanobacteria. If you have any doubt, please ask me. Tomorrow class we will going to discuss about Nostoc, Anabina, Oscillatoria. Tomorrow and day after tomorrow, we will going to complete Nostoc, Anabina, Oscillatoria. These are the type studies of the blue-green algae. My dear friends, if you have any doubt, please ask me. I will clarify.
Girls, if you any doubt to ask the sir, ma. Girls, if you no doubt, ma, it's clear.
No,
I
ma'am.
want to ask the
Because they are very
questions
good-
tomorrow
they are very
class.
good students.
Clear,
This
ma'am.
topic.
Okay,
Yes,
ma'am.
they are ready because they are very keenly observed.
Okay,
They are
ma'am.
very, very interestingly they are listen because they are the good students. Ready college students are always good students and sincere students to listen carefully. And whenever we will going to ask, they are ready to answer today's topic. Am I right, my dear friends?
Okay, sir. Thank you so much.
Sir.
Very good.
Girls, both combination girls to take the attendance also, ma. Okay.
Okay, ma'am.
Okay, ma'am.
Thank you, sir.
Thank you,
Thank you,
sir.
sir.
Thank you, sir.
Thank you, sir.
Sir.
Okay, girls. Good night. See you tomorrow.
Thank you, sir.
Thank you, ma'am. Good.
Good night, ma'am.
Good night.
Good night, ma'am.
Good night, ma'am.
Good night. Good night. Good